Welcome back to Simplifying Synthesis. In this video, we are going to look at Prodrug Strategies against the Varicella Zoster Virus. This work was published by the groups of Singh and Chu in the Journal of Medicinal Chemistry. The Varicella Zoster Virus is very common and causes chickenpox in the primary infection, most commonly in children. After the primary infection, the virus remains dormant and can reactivate later in life, where it causes shingles. While vaccination efforts have been quite effective, it is estimated that there are still over 1 million cases annually in the US, with the most at-risk groups being people over the age of 50, transplant recipients, and people with HIV. The antivirals currently used include acyclovir, valcyclovir, and famiciclovir. While these drugs do work, they are not particularly effective against VZV, requiring large doses, and long-term use of these compounds is associated with the development of drug resistance. Due to these issues, there is a great need for better drugs for this virus. A common strategy in the design of antiviral drugs is to utilize chain-terminating nucleoside analogues. During DNA replication, new nucleosides are incorporated into the growing DNA strand by sugar phosphate bonds at the 3 and 5 positions. Chain-terminating antivirals mimic these naturally occurring nucleosides and are able to be incorporated into the growing chain. However, these lack a 3' hydroxyl group, so the chain cannot grow any further, and the DNA replication is terminated. The compound that is the focus of this paper, L-BHDU, has previously been reported by this group and was shown to have an EC50 value of 0.25 micromolar with a selectivity index of over 900. This compound mimics the natural nucleoside deoxythymidine and utilizes the same nucleobase as brivudine, a compound that has potent antiviral activity but shows undesired side effects when used in combination with other drugs. In the previous studies, the researchers found that they could increase the antiviral activity of LBHDU by synthesizing a valine ester prodrug. In this work, they further expand on this concept by synthesizing a range of amino acid prodrugs, including the unnatural D-isomer of valine. They also explore phosphoramidate type prodrugs, which are also known as ProTides, which is short for prodrug of a nucleotide. These prodrugs are first activated by cathepsin A, which is an esterase enzyme. The carboxylate generated by this cleavage can then undergo an intramolecular attack on the phosphoramidate, forming a 5-membered ring upon the elimination of the phenoxide. This intermediate can then be attacked by water, either directly at the phosphorus, as shown here, or also at the ester. This hydrolysis forms a phosphoramide, which is then cleaved by a phosphoramidase to form the BHDU monophosphate. This type of prodrug was first introduced by McGuigan in the 90s and has been widely used in antiviral drug design since. These are quite attractive, as nucleoside mimetics must first be phosphorylated before they can be incorporated into a growing DNA strand. In vivo, it is the first phosphorylation event that is the rate-limiting step in this process, so by incorporating a prodrug moiety that produces a monophosphate upon activation, it can drastically increase the effectiveness of the drug. In these studies, they also utilize long-chain phospholipid prodrugs. These increase the hydrophobicity of the molecule and can improve the pharmacokinetic properties. Like phosphoramidates, they can be cleaved in vivo to generate the monophosphate. Similarly to these long-chain phospholipids, the bis-POM and bis-POC prodrugs also increase the lipophilicity of the molecules and can be hydrolyzed to generate the monophosphorylated drug. The initial studies on these compounds looked at the antiviral activity and also the cytotoxicity. These studies were done in ARPE19 cells. These cells were infected with the strain of VZV that expresses firefly luciferase. This allows for the virus spread to be measured using bioluminescence imaging. From these studies, we can see that the amino acid prodrugs mostly showed similar bioactivity, with very low cytotoxicity and good antiviral activity. One notable observation is the much reduced antiviral activity of the compound containing D-valine. This is the reason the authors did not explore any other D amino acids. The compounds incorporating the ProTide moiety showed low cytotoxicity, but also much reduced antiviral activity. In contrast, the phospholipid compounds showed moderate antiviral activity, but also greater cytotoxicity. This could possibly be due to a slower metabolism of these compounds. Finally, the phosphate ester compounds showed both good antiviral activity and also very low cytotoxicity, with selectivity indices of almost 3000. These results are much better than those for acyclovir and cidofovir, which were used as positive controls, so these compounds were selected for further study. In the next experiments, the compounds were tested against both wild-type and mutant strains of the virus. These mutants include a thymidine kinase knockout. Thymidine kinase catalyzes the phosphorylation of deoxythymidine to deoxythymidine monophosphate. This enzyme is quite important in antiviral drug discovery, as the viral thymidine kinase has a different specificity to the human enzyme, and this difference can be exploited to design drugs that are more targeted towards the viral enzyme and therefore increase selectivity. From these studies, we can see that the prodrugs were ineffective in the viral strains where the TK enzyme was knocked out. This indicates that the thymidine kinase enzyme is essential to their mode of action. In these studies, they also investigated a thymidylate synthase knockout. Thymidylate synthase is an enzyme that catalyzes the conversion of deoxyuridine monophosphate to deoxythymidine monophosphate, providing an alternate source of D-TMP in the cell. All of the compounds screened showed a greater efficacy in the thymidylate synthase knockouts, and this is attributed to the reduction of the D-TMP in the cell, resulting in less competition with the LBHDU prodrug for incorporation into the DNA strand during the viral DNA synthesis. With the antiviral activity now determined, they then investigated the pharmacokinetic properties of these compounds. Using the pivyloyl oxymethyl prodrug, or POM, they incubated it with homogenized mouse liver and tracked the generation of LBHDU over time. These studies showed that the prodrug is rapidly hydrolyzed, showing a maximum concentration of the LBHDU monophosphate in just 4 minutes. Interestingly, the non-phosphorylated LBHDU was also formed, indicating that the hydrolysis also occurs at the 5' position of the dioxylane ring. To see how these compounds behave in vivo, both the L-BHDU drug and the POM prodrug were administered to mice by oral gavage, and blood was collected at 20-minute intervals. LCMS was then used to determine the drug concentration in the blood plasma. These experiments show that the prodrug has a much higher bioavailability than the parent L-BHDU. The monophosphate was also detected, but the concentration was below the limit of quantitation. This suggests that the monophosphate is rapidly cleaved at the 5' phosphorus-oxygen bond to release L-BHDU. Taken together, these biological studies show that L-BHDU is a very potent antiviral that is well tolerated by cells in vitro and also has very favourable pharmacokinetic properties in vivo. While they did not report any antiviral studies in vivo, it is very likely that these compounds will show good activity in an animal model and may have the potential to make it into the clinic. So with the biology out of the way, let's look at the synthesis. This starts with 1,6-anhydromannose, which is first reacted with dimethoxyacetone and PTSA. This selectively forms an acetonide between the 2 and 3 hydroxyl groups, as these are on the same face of the ring, and therefore is much easier to form than the acetonide between the 3 and 4 position. The hydroxyl group on the 4 position can then be protected as a benzyl group, and the acetonide is then hydrolyzed using sulfuric acid. With this syn diol now revealed, they could carry out an oxidative cleavage using sodium periodate. This first forms an adduct with the diol, rapidly oxidizing it, breaking the carbon-carbon bond, and forming 2 aldehydes. These aldehydes were then reduced using sodium borohydride. Interestingly, they observed the migration of the benzyl group during this reaction, leaving it residing on the primary hydroxyl group of the alkyl chain. Taking this compound forward, the remaining primary hydroxyl group was then protected using TBDPS. This protecting group is very large and therefore was selective for the more accessible primary hydroxyl group. With this in place, the benzyl group was then hydrolyzed with sodium methoxide. This hydrolysis set the stage for another oxidative cleavage of a diol, this time using ruthenium dioxide in addition to sodium periodate. The combination of these reagents generates ruthenium tetroxide in situ, which is a very powerful oxidizing agent. It first cleaves the diol, forming an aldehyde, similar to the mechanism we saw before, and then oxidizes the aldehyde further, forming the carboxylic acid. This acid then took part in an oxidative decarboxylation. The compound is reacted with lead tetraacetate, where it displaces one of these acetate ligands and coordinates to the lead. This then undergoes a decarboxylation reaction, generating a radical on the dioxylane ring. This is further oxidized by lead tetraacetate, forming an oxonium ion that then undergoes nucleophilic attack from one of the acetates present in solution. This generated the acetylated product as a mixture of isomers. This acetate was required for a Vorbruggen glycosylation. It is first reacted with TMS iodide, which silates the carbonyl and promotes its elimination, generating an oxonium intermediate. TBS triflate is added to the reaction, and this reacts with the bromovinyl uracil, silating both of the carbonyl oxygens. The iodide, present in solution, can then attack these silyl groups, promoting the nucleophilic attack from the nitrogen towards the oxonium center. This generated the nucleoside as a mixture of isomers, and the TB-DPS group was deprotected using TBAF to produce L-BHDU. To generate the ProTide, phenyl dichlorophosphate was first reacted with L-alanine isopropyl ester, eliminating the chloride and forming a phosphorus-nitrogen bond. The reaction with L-BHDU eliminates the remaining chloride and completes the synthesis of the phosphoramidate prodrug. Well, that's everything for this video. Join me in the next one, where we will look at the total synthesis of Eugenial C.